Right now, inside every part of your body, the most incredible performance is taking place. It's happening millions of times a second. It's really the most essential drama in all of biology. And it's the story of how one single cell becomes two. So this isn't just some dry biological process. It's the cell's great divide, a spectacular performance in 4 acts.
So here's our program for today. We're going to start by setting the stage for this whole dramatic performance. Then we'll walk through the 3 main acts of this cellular play, and we'll finish with a final curtain call, where we get to see the grand result of this whole intricate show.
So this really is the central question, isn't it? I mean, how does your body grow? How does a cut on your skin heal itself? It all comes down to this absolutely incredible ability for one cell to divide and create a perfect identical copy of itself. And the answer to that question? It's a process called mitosis. Now that phrase right there, equational division, is so important. It means mitosis isn't about cutting things in half or reducing anything. Nope. It's about a perfect duplication. The number of chromosomes in the parent cell and in the 2 new daughter cells stays exactly the same. You can think of it as biological copy and paste, and it is at its absolute finest. Our source material actually calls this the most dramatic period of the cell cycle. And you know what? It's easy to see why. This is not some minor tweak. We're talking about a massive reorganization of pretty much every single component inside the cell. It's a high-stakes, perfectly choreographed event.
And just like any great play, the division of the nucleus, which has a fancy name, Karyokinesis, is broken down into acts. And these are the 4 main stages we're going to follow: Prophase, Metaphase, Anaphase, and Telophase. Okay, let's take a look at each act.
Welcome to Act 1, Prophase. Before the main event can even start, the cell has a ton of prep work to do. This whole phase is about intense preparation, where the main characters, the chromosomes, have to get into costume and all the backstage machinery has to be set up perfectly.
And this is really the star moment of Prophase. All of those long, tangled-up threads of chromosomal material start to condense. They wind themselves up super tightly, becoming these compact little structures that we can actually see under a microscope for the first time. They are totally getting ready for their moment in the spotlight.
And what's just wild is that all of this is happening at the same time. As the chromosomes condense, each one pops into view looking like that classic X shape. And that X is made of 2 identical copies, which we call sister chromatids, and they're held together right in the middle at a point called the centromere. Meanwhile, these little structures called centrosomes, think of them as our stagehands. They start moving to opposite sides of the cell and begin building the mitotic spindle. And to clear the stage, the nuclear envelope just dissolves.
Okay, let's move on to Act 2 and see how this all builds. I mean, this is the climax of our performance, right? These are the spectacular, breathtakingly precise moments of Metaphase and Anaphase. This is where the genetic material gets sorted, and the fate of the 2 future cells is literally decided.
This moment right here is metaphase. The nuclear envelope is gone. The chromosomes are all dressed up in their costumes, and now they're moved by these spindle fibers right to the dead center of the cell. They line up in a perfect, single-file line. It creates this moment of incredible tension right before the big separation.
And this just highlights the incredible precision that's involved. Remember those sister chromatids? Well, spindle fibers from one side of the cell grab onto one sister, and fibers from the other side grab onto the other sister. It's this perfect tug-of-war that lines them all up at the cell's equator, forming what we call the metaphase plate. It makes sure the next step is perfectly fair. And then, the action begins. Anaphase kicks off with this single, powerful event. That centromere holding each pair of sister chromatids together, it splits, and it happens simultaneously across every single chromosome.
And just like that, in an instant, the connection is broken. The sister chromatids aren't sisters anymore. They are now their own individual daughter chromosomes, and they start this rapid journey to opposite ends of the cell.
And this is it's a brilliant illustration of the mechanical genius of the cell. The spindle fibers literally pull the newly separated chromosomes apart. You can almost picture it, right? The centromere is leading the charge toward the pole, and the arms of the chromosome are trailing behind it like streamers. It's this beautiful, coordinated retreat to safety.
So this brings us to the final act, telophase, and the grand finale, cytokinesis. The drama of separation is over, and now it is time for the resolution. The cell has done the incredibly hard work of separating its DNA. Now it just has to finish the job and form two brand new, fully functional cells.
This part is telophase. The daughter chromosomes have finished their journey. They've arrived at opposite poles of the cell. Their big performance is over, so now they can relax and, well, take their final bow.
You know, a really great way to think about telophase is just prophase in reverse. The chromosomes uncoil and kind of fade from view. A brand new nuclear envelope forms around each of the two sets of chromosomes, creating two perfect daughter nuclei. The cell's command centers are officially rebuilt.
But hold on, we're not quite done yet. We've got two nuclei, which is great, but they're still hanging out inside one big cell. So the final step is something called cytokinesis, and that's where the cytoplasm, the stage itself, splits right down the middle to complete the process.
And how this split happens actually depends on the cell type. So in animal cells, a little furrow forms and basically pinches the cell in two, kind of like tightening a drawstring on a bag. But plant cells have that rigid cell wall, right? So they can't do that. Instead, they build a new wall called a cell plate right down the middle, and it grows outward to divide the cell. Two totally different methods, but they both lead to the same perfect result.
And with that, the performance is complete. The curtain falls, and where there was once one cell, there are now two. So let's take one final look at what this amazing process has accomplished and why it is so incredibly fundamental to, well, all life. This quote from our source really just says it all, doesn't it? Mitosis is a two-part victory. First, it perfectly separates the genetic blueprint into two brand new nuclei. And second, it divides the entire cell, making sure that each new cell has everything it needs to start its own life cycle.
And this is the ultimate takeaway. All of that drama, the condensing, the lining up, the splitting, the rebuilding, all of it serves one single purpose: to turn one cell into two genetically identical daughter cells, ready to grow, function, and then one day perform this very same dance all over again.
It's an unbelievably robust and accurate process, but it is worth thinking about. What happens when a step gets missed? What happens when the choreography fails? The incredible precision of mitosis is the very basis of healthy life, which leaves us with a final and pretty crucial question: what are the consequences when this perfect performance goes wrong?